They're too small to see with the naked eye, but these microbes have abilities that put superheroes to shame.
Chemolithotrophs.
Your name is Frankie Nodatum. Press confirm if you are ready to play.
Imagine you are a chemolithotroph, a bacterium that can utilize an inorganic compound as electron donor for metabolism. You will use it to produce energy in the form of ATP. In chemolithotrophs, the compounds, the electron donors, are oxidized in the cell, and the electrons are channeled into respiratory chains, ultimately producing ATP. What inorganic compound will you choose?
Choose your fighter, inorganic compound. You are a hydrogen oxidizing bacterium. You will be accompanied by your comrades, your fellow hydrogen oxidizing bacteria.
You utilize hydrogen as an energy source aerobically. This process is called hydrogen oxidation, wherein hydrogen serves as an electron donor and is oxidized by a membrane-bound hydrogenase, causing proton pumping via electron transfer to various kinones and cytochromes.
You are now entering hydrogen oxidation dungeon.
Specifically, the electrons from hydrogen are catalyzed by the membrane-integrated hydrogenase and are initially transferred to a kinone acceptor. The electron then travels to a series of cytochromes, and the oxygen serves as the terminal electron acceptor. The generated proton motive force from the electron transport will eventually reduce oxygen to water. Also, the electron flow allows the protons to be pumped out of you and will create a high concentration of protons outside you. The protons will be transported back inside the membrane through the ATP synthase complex and will result in the formation of ATP. Some of you can synthesize two hydrogenase enzymes. First, the membrane-integrated, and second, the cytoplasmic hydrogenase. The cytoplasmic hydrogenase catalyzes the direct reduction of NAD+ to NADH, which is then used to fix carbon dioxide via Calvin cycle. Most of your kind grow best under microaerophilic conditions because the hydrogenase enzyme is inhibited by oxygen. But oxygen is still needed for it to serve as the terminal electron acceptor.
Well done. You have now gained the ability to oxidize hydrogen.
What would you like to do next?
You pressed sulfur. You are a sulfur oxidizing bacterium. You will be accompanied by your comrades, your fellow sulfur oxidizing bacteria,
the Beggiatoa and Paracoccus bacteria. Sulfur oxidation happens when the hydrogen sulfide within you donates its electron to a flavin mononucleotide such as FAD, which serves as the starting point of the oxidation process. The electrons in FAD are transferred to cytochrome BC1 complex by kinone, which is a mobile electron carrier. Electrons from cytochrome BC1 are then transferred to cytochrome AA3 complex through another mobile electron carrier, the cytochrome C. Lastly, the electron is received by the final electron acceptor of the process, that is the oxygen. Then oxygen will be reduced to water. Other reduced sulfur compounds, such as elemental sulfur and thiosulfate, can also donate their electrons in cytochrome C and still follow the same process. During the electron transport, hydrogen ions are pumped outside you, which results in the formation of proton gradient and will then drive a proton motive force that will translocate the protons via ATP synthase complex and will lead to the formation of ATP. The excess ATP produced is used to fuel the reverse electron flow from FAD to NAD+, that will result in the reduction of NAD+ to NADH. The NADH will then produce ATP and fix carbon dioxide for you to produce a cellular material.
Well done. You now gained the ability to oxidize sulfur.
What would you like to do next?
You pressed iron. You are an iron oxidizing bacterium. You will be accompanied by your comrades, your fellow iron oxidizing bacteria.
There are three types of them. We have the acidophiles, those that thrive in an environment with a very low pH. We have microbes at circumneutral pH, the microaerophiles found in oxic-anoxic environments. And lastly, we have the anaerobic photosynthetic bacteria, those that do not produce oxygen during photosynthesis. The electron from iron 2 ion is transported to rusticyanin, a vital complex used by your kind. This rusticyanin will then transfer the electron to either the first electron complex, the cytochrome Bc1, or to the electron carrier, the cytochrome C. The electron from cytochrome Bc1 is transferred to cytochrome AA3 through the cytochrome C. The electron will then be accepted by the final electron acceptor, which is oxygen, that is reduced to form water. The electron flow will then allow protons to be pumped out of you and will create a high concentration of protons outside you. The protons will be transported back inside your membrane through the ATP synthase complex and will result in the formation of ATP. Excess ATP will allow reverse electron flow to occur. The electron from cytochrome Bc1 will be transferred to NAD+ through quinone, producing NADH from the reaction of NAD+. NADH will then generate ATP and fix carbon dioxide into your cellular material.
Well done. You now gain the ability to oxidize iron.
What would you like to do next?
You pressed nitrogen. You are a nitrogen oxidizing bacterium.
In your journey, you are involved in a cycle called nitrogen cycle with different processes. In this cycle, other organisms are involved as well, but they will be found in processes such as assimilation, ammonification, and denitrification. You, however, play a key role in nitrogen fixation and nitrification, or ammonia oxidation and nitride oxidation, respectively, and anoxic ammonia oxidation. Choose the process you want to get involved in. In your journey, you will be accompanied by your comrades, your fellow nitrogen oxidizing bacteria involved in nitrogen fixation. You will be given a special power enzyme called nitrogenase, but these enzymes can get inactivated by oxygen or oxygen gas. Your comrades have their own ways to protect it. Symbiotic nitrogen-fixing organisms. They create a symbiotic relationship with plants. Plants get ammonia from them while plants give them ATP and protection so that nitrogenase enzymes can function well. Free-living nitrogen-fixing organisms. They are the cyanobacteria. They do not need to make a symbiotic relationship with plants in order to have a good environment for the nitrogenase enzyme. Instead, they have specialized cells known as heterocysts. Inside the heterocyst is where the process of fixing the nitrogen happens. Nitrogen is an essential component in many important biomolecules such as amino acids and nucleotides. One major purpose of nitrogen metabolism is to convert nitrogen or dinitrogen gas into a usable form. This metabolism process needs electrons and energy and is done by either lightning or the more relevant and important nitrogen-fixing bacteria, specifically called the diazotrophs. These microorganisms have an enzyme called the nitrogenase that is responsible for nitrogen fixation. However, this enzyme can be deactivated by oxygen. That is why this process must occur in an anaerobic environment. The mechanism of the nitrogen metabolism process happens in a complex of enzymes called the nitrogenase complex. This complex converts nitrogen molecule into two ammonium ions. The nitrogen molecule, with the addition of 6 electrons and 8 hydrogen ions, react to form 2 ammonium ions. The ammonium ions are already usable in important bioprocesses. The overall reaction would be dinitrogen plus 8 electrons plus 16 ATP plus 10 hydrogen ions produces 2 ammonium ions plus 16 ADP plus 16 inorganic phosphate and hydrogen gas. The 16 ATP is provided by the plant, while the produced ammonium ions can now be used for the biosynthesis of amino acids and nucleotides. The reaction in the nitrogenous complex presents only 6 electrons and 8 hydrogen ions, while the overall reaction has 8 electrons and 10 hydrogen ions. It is because the overall net reaction presented that the hydrogen gas consumed the 2 electrons and 2 hydrogen ions from the reactants. Congratulations! You can now fix nitrogen. Would you like to proceed to nitrification?
You pressed nitrification.
In your journey, you will be accompanied by your comrades, your fellow ammonia oxidizing bacteria, and the nitrite oxidizing bacteria involved in nitrification. These are soil bacteria, Nitrosomonas, Nitrococcus, and Nitrobacter. Nitrification is a two-step process in which ammonium or ammonia ion is converted to nitrate ion. First, your comrades Nitrosomonas and Nitrococcus convert ammonium to nitrite ion, and then Nitrobacter oxidizes nitrite ion to nitrate ion. You gain energy through these conversions, both of which require oxygen to occur. Step 1. In ammonia oxidizing bacteria, ammonium is oxidized by the enzyme ammonia monooxygenase that produces hydroxylamine and water. The second essential enzyme, hydroxylamine oxidoreductase, oxidizes hydroxylamine to make the product nitrite ion. 4 electrons are removed in the process. Ammonia monooxygenase is an integral membrane protein, whereas hydroxylamine oxidoreductase is a periplasmic protein. In the reaction mediated by ammonia monooxygenase, 2 electrons and 2 protons are utilized to reduce 1 atom of oxygen to water. These electrons come from the oxidation of hydroxylamine and are supplied to ammonia monooxygenase from hydroxylamine oxidoreductase via cytochrome C and ubiquinone. Thus, for every 4 electrons generated from the oxidation of ammonium to nitrite ion, only 2 actually reach cytochrome AA3, the cytochrome that interacts with oxygen to form water and yield energy. Step 2. On the other hand, nitrite oxidizing bacteria utilize the nitrite oxidoreductase enzyme to oxidize nitrite ion into nitrate ion. Due to the high potential of the nitrate-nitrite couple, the electrons travel a very short electron transport chain to the terminal oxidase. Cytochromes of the A and C types are present in the electron transport chain of the nitrite oxidizers, and the activity of cytochromes AA3 creates a proton motive force. Only small amounts of energy are available in this reaction. This is why the growth yields of nitrifying bacteria are low.
Congratulations! You can now oxidize both ammonia and nitrite.
Would you like to proceed to anoxic ammonia oxidation or ANAMOX? In your journey, you will be accompanied by your comrades, your fellow ANAMOX bacteria, Brocadia anamoxidans, which is a marine bacteria. Your other comrades are Coninia, Anamoxyglobus, Jetinia, and Scalindua. In anoxic ammonia oxidation, nitrogen compounds are utilized as both electron acceptor and electron donor. Ammonia is oxidized anaerobically as the electron donor, while nitrite is used as the electron acceptor, with the dinitrogen gas produced as a byproduct. The reactions occur within a specialized cytoplasmic structure which constitutes 50 to 70% of the total cell volume, the anamoxosome. The strong anamoxosome membrane is required to protect the cell from the toxic intermediates produced during anamox reactions. These include, in particular, the compound hydrazine, a very strong reductant. In anamox reaction, nitrite ion is first reduced to nitric oxide by nitrite reductase, and then nitric oxide reacts with ammonium to yield hydrazine by activity of the enzyme hydrazine hydrolase. Hydrazine is then oxidized to dinitrogen plus electrons by the enzyme hydrazine dehydrogenase. Some of the electrons generated at this step enter the anamoxosome electron transport chain that yields a proton motive force and ATP by ATP synthase, while others feed back into the system to drive the electron-consuming earlier steps. Do you want to see how assimilation, ammonification, and denitrification work? Assimilation is the process by which plants and animals incorporate the nitrate ion and ammonia formed through nitrogen fixation and nitrification. Plants take up these forms of nitrogen through their roots and incorporate them into plant proteins and nucleic acids. Animals are then able to utilize nitrogen from the plant tissues. Ammonification is the conversion of organic nitrogen into ammonia. The ammonia produced by this process is excreted into the environment and is then available for either nitrification or assimilation. Denitrification is the reduction of nitrate ion to gaseous dinitrogen by anaerobic bacteria. This process only occurs where there is little to no oxygen, such as deep in the soil near the water table. Hence, areas such as wetlands provide a valuable place for reducing excess nitrogen levels via denitrification processes.
Congratulations! You now know how assimilation, ammonification, and denitrification work.
Well done! You gained the ability to oxidize nitrogen. Wow! What amazing journeys you had! Do you want to look back on some of your most important moments? You are a lithotroph, an organism that obtains its energy from inorganic compounds such as ammonia via electron transfer. Specifically, you belong to the chemolithotrophs, bacteria which utilize different inorganic compounds as electron donors to produce energy in the form of ATP. The most common substances are hydrogen gas, sulfur compounds such as sulfite and sulfur, nitrogen compounds such as ammonium and nitrite, and lastly, ferrous iron. Hydrogen oxidizers are organisms which oxidize hydrogen gas with the use of a hydrogenase enzyme. Both aerobic and anaerobic hydrogen oxidizers exist, with the aerobic organisms eventually reducing oxygen to water. Sulfur oxidizers. As a group, these organisms are capable of oxidizing a wide variety of reduced and partially reduced sulfur compounds such as hydrogen sulfide, elemental sulfur, thiosulfate, and sulfite. Sulfate is frequently a byproduct of the oxidation. Often, the oxidation occurs in a stepwise fashion with the help of the sulfite oxidase enzyme. Nitrogen oxidizers. The oxidation of ammonia is performed as a two-step process by nitrifying microbes, where one group oxidizes ammonia to nitrite and the second group oxidizes the nitrite to nitrate. The entire process is known as nitrification and is performed by small groups of aerobic bacteria and archaea, often found living together in soil or in water systems. Iron oxidizers. These organisms oxidize ferrous iron to ferric iron. Since ferrous iron has such a positive standard reduction potential, the bioenergetics are not extremely favorable, even using oxygen as a final electron acceptor. This situation is made more difficult for these organisms by the fact that ferrous iron spontaneously oxidizes to ferric iron in the presence of oxygen. The organisms must use it for their own purposes before that happens. You completed all possible journeys. You have unlocked the bonus quest. You are a bacterium capable of metabolizing inorganic compounds. You and your fellow bacteria are being researched for your potential impacts in the biosphere, mainly for the renewal of your environment and maintenance of biogeochemical cycles. This is to ensure the constant availability of elements in the environment, to keep the balance in your ecosystem, and to reduce potent damaging human wastes. For you, the inorganic oxidizers, it is almost often the latter case.
The contributions of inorganic oxidizing microorganisms in shaping the biosphere. Do you want to proceed to your quest? You pressed biodegradation.
The presence of toxic compounds in the environment prompted scientists to look into your biodegrading properties. Biodegradation mainly aims to recycle wastes, break down organic matter into useful nutrients, and return them to the environment, all through biological processes by living microorganisms such as you. This allows pollutants such as pesticides, dyes, and radionuclides to degrade more naturally. You can degrade or biotransform some of these pollutants, such as polychlorinated biphenyls, or PCBs, which are commonly used in electrical and heat transfer equipment, paint, plastics and rubber products, and carbonless copy papers. Anaerobic bacteria subject higher chlorinated PCBs to reductive dehalogenation, while aerobic bacteria oxidize lower chlorinated biphenyls. The aerobic pathway is catalyzed by enzymes, which are biphenyl dioxygenase, dihydrodiol dehydrogenase, 2,3-dihydroxybiphenyl dioxygenase, and hydrolase. The capable bacteria are mostly gram-negative, such as Pseudomonas and Comamonas, but may include gram-positive as well, such as Bacillus and Microbacterium. Then there are the heavy metals. Because heavy metals cannot be destroyed, they must be converted to a stable form or removed. Thus, you act on these metals through biotransformation. There are mechanisms that come into play in dealing with this kind of pollutant. Biosorption is the metal sorption to the cell surface by physiochemical mechanisms. Bioleaching is heavy metal mobilization through excretion of organic acids or methylation reactions. Biomineralization is heavy metal immobilization or precipitation through formation of insoluble sulfides or polymeric complexes by sulfate-reducing bacteria. Finally, intracellular accumulation and enzyme-catalyzed transformation through redox reactions complete the biotransformation of heavy metals. You and your comrades have also developed resistance mechanisms against toxicity to aid you in biotransforming heavy metals. Here are some of them. Reduction of metals through dissimilatory metal reduction. Utilizing metals as terminal electron acceptors in anaerobic respiration. Reduction mechanisms independent from respiration that impart metal resistance, such as chromium-6 to chromium-3, selenium-6 to elemental selenium, uranium-6 to uranium-4, mercury-2 to mercury-0. Microbial methylation, potent in bioremediation as methylated compounds are frequently volatile, like mercury-2 to gaseous methylmercury by Bacillus or Pseudomonas aeruginosa. Acidophilic iron bacteria, such as Acidithiobacillus ferrooxidans and sulfur-oxidizing bacteria, leach high concentrations of astatine, cadmium, copper, cobalt, and zinc from contaminated soils. Active uptake of metals compared to biosorption or passive uptake. Metals pass into the cell across cell membrane through cell metabolic cycle. like Pseudomonas for removal of toxic radionuclides such as uranium-6 and thorium-4 from nuclear waste streams. Genetic engineering on some of your comrades has sparked great interest. The chromosomally located genes for PCB catabolism from Aureutropha, a chromobacter genus, and A. denitrificans were transferred into a heavy metal-resistant strain of Aureutropha through natural conjugation. Biodegradation is a crucial tool in the overall goal of bioremediation, wherein you are used to remove contaminants, pollutants, and toxins from soil, water, and other environments. It is a growing branch of biotechnology, and sadly but truly, one where you, microorganisms, are responsible to clean up the mess we humans have made.
Bonus quest. Well done. You completed biodegradation. Press any key to continue.
Biomining. Biomining is the process of using you to catalyze the extraction of minerals. These minerals can be economically valuable, such as rock ore, copper, and gold. They can also be mine waste from cleaning up sites that are polluted with metals. Currently, it is used to produce about 5% of gold and 10 to 20% of copper found in the world. Biomining mostly targets the economically valuable metals that are commonly found in sulfidic minerals. Some of you, usually acidophilic iron and sulfur oxidizers, oxidize these minerals, allowing them to dissolve in water. These metals are more easily recovered this way compared to solid rocks. Heterotrophic bacteria can also extract minerals in circumneutral or alkaline pH by changing the local environment pH, often by the release of protons or organic acids. This is bioleaching, when the metal of interest is directly dissolved and is applied to base metals. For example, iron and copper are converted into forms that dissolve more easily. It basically exploits your ability to obtain metals by mixing them in solution with ores and collecting the metal as it floats to the top. The most common processes in biomining are heap leaching, where freshly mined materials are moved directly into heaps that are then bioleached; dump leaching, where low-value ores or waste rocks are placed in a seal pit and then bioleached. This is to extract more of the valuable metals from the waste pile. Agitated leaching, where crushed rocks are placed in a large vat that is shaken to distribute yourselves who are also inside the vat and the material evenly, and is used for some copper sulfides like chalcopyrite. This speeds up the bioleaching process. Leaching can take days to months, which is slower compared to conventional mining techniques. Another method is biooxidation, when the metal of interest is made more accessible or enriched in the material left behind. It is normally applied to the sulfidic refractory gold ores and concentrates. This means that gold, for example, is not directly dissolved but is made more accessible to traditional mining techniques as the minerals surrounding it are dissolved. Biomining also has its risks. The metal-rich acids you excrete tend to leak, similar to acid mine drainage. This can be properly managed, however, by ensuring that biomining is conducted in controlled conditions with proper sealing and waste management protocols. In that case, these risks are significantly minimized compared to the conventional mining methods. For one, it can reduce the environmentally damaging use of toxic compounds such as cyanide. The risks of releasing your kind into the local environment are small, as they are already abundant on Earth itself. It is cheaper and more simple, and it can effectively extract metals from low-grade sources. Along with base earth metals, this process also seeks out to extract rare earth elements, or REEs, buried deep in the surface of the Earth that are essential for lots of modern technologies, such as electronics. It has also shown potential in outer space. Studies conducted in the International Space Station demonstrated some of you mining economically important elements in REEs. One such study involved Sphingomonas dessicabilis, Bacillus sp, and Cupriavidus metallidurans bioleaching on basaltic rock, in microgravity and Mars gravity. Further developments on this field becomes a larger step in space mining and advanced human industry beyond Earth. In the future, biomining might become a more common practice. Other techniques are currently being developed, including large-scale biomining, recycling wastewater elements, recovering metals from acid mine drainage, and deep underground biomining.
Bonus quest. Well done. You completed biomining.
You are now a fully grown chemolithotroph. Go forth, perform your duties, and fulfill your biological destiny.